Good morning, student. I am Mr. Vinod Thomri, and we are studying cytogenetics. Our today's topic in cytogenetics is the introduction of the pioneer of cytogenetics, that is the Mendel. We, that we call them as a father of genetics due to his important work in genetics. Mendel, the life period is 1822 to 1884. He conducted breeding experiment in garden pea plants. The botanical name of pea plant is Pisum sativum. Studied the individual pea plant in many generations. He put forth basic laws of heredity called as Mendelism and also called Mendel's law of heredity or inheritance. His work is well known and called as Mendelism. Therefore, Mendel is called as a father of genetics. As we mentioned, Mendel studied several characters in pea plants. The selected characters from his study in Mendelian experiments are as follow. The above rows, this shows the dominant character, and the below rows which shows the recessive character which study by the Mendel. The first character he study is seed shape. In case of dominant character, you will found seeds with round in shape, and in case of recessive character, the seeds with wrinkle shape. In case of seed color, dominant seed color in pea plant is yellow and the recessive color is green. In case of pod shape, the inflated shape is the dominant character in case of pea plant, and the constricted shape of pod is the recessive character in pea plant. In case of pod color, the green color of pod is the dominant character and the yellow color of pod in pea plant is a recessive character. In case of flower color in pea plant, the purple color flower is the dominant trait or the character, and the white color flower is the recessive trait or character in case of pea plant. The flower location in pea plant, the dominant character is axial position of the flower and the recessive character is terminal position of the flower. Then the plant size, the tall plant is the dominant character in case of pea plant, and the dwarf size of the plant is the recessive character in case of pea plant. By using this character or this study, Mendel put forth some laws. The first law he put forth is law of dominance. The statement of this law is the law of dominance says that when an organism is heterozygous for a trait, only the dominant allele will produce a phenotype. Heterozygous condition means the genotype is like this: one is in dominant form and one is in recessive form. In such case, always the dominant form or the dominant allele will express in the F1 generation. For example, when Mendel crossed a pure breeding tall pea plant with pure breeding dwarf pea plant, in F1 generation all the progenies are tall due to the expression of dominant allele, that is the capital T. Let's see this with the example. Here one male and one female plant, they are cross. The female plant with phenotype tall and the male plant with phenotype dwarf, they are selected for crossing. The genotype of tall plant is capital T, and the genotype of dwarf pea plant is small t. Then the gametes of tall plant out of capital T, one capital T is involved for the fusion and fertilization process, and from tall capital T and from dwarf there is a one small t gamete will take part in the fusion process and fertilize to form a next plant. When fertilization occurs between the due to the fusion of those two gametes, the F1 generation is with all the tall plant because the dominant allele, that is capital T, masks the expression of recessive allele, that is small t. This condition is called heterozygous condition, and in heterozygous condition the dominant allele always shows his phenotypic character. Then the next law which put forth by Mendel is law of segregation, or which is also called as law of purity of gametes. The statement of this law is, the law states that two contrasting characters, though they remain together in F1 hybrid, they segregate or separate during the formation of gametes. After segregation of F1 progeny, that is hybrid, each gamete or allele is 100% pure for the characters which it carries. That is, in case of plant, height trait. In case of plant, height trait, which is expressed as a capital T allele and small t allele. Here, capital T is 100% pure for tallness, while small t is 100% pure for dwarfness. Hence, law of segregation, also known as a purity of gamete. Law of purity of gametes. Now, this is the explanation of this law. When we cross two parent plants P1 and P2, one is selected as a female plant and one is selected as a male plant, when with the phenotype tall and dwarf. The genotype of tall pea plant is capital T and small male pea plant small t. When we proceed for the gamete formation, the small t and capital T, they fuse together and fertilize to form a F1 generation and all the plants with tall height in heterozygous condition, F1 hybrid heterozygous. When for the when we proceed for the formation of gametes of F1, then when capital T and small t they segregate from each other, they shows the 100% their phenotype when they cross with another plant. That is, capital T shows 100% tallness character, and small t when crossed with small t they shows the dwarfness, 100% dwarfness. Then the third law, that is the law of independent assortment. The statement of this law is, Mendel's law of independent assortment states that the allele of two or more different genes get sorted into gametes independently of one another. In other words, the allele a gamete receives for one gene does not influence the allele received for another gene. For explanation of this, there is a requirement of dihybrid cross. Mendel crossed a pure breeding tall pea plant having red flower with a pure breeding dwarf pea plant having white flower. In F1 generation, all the progenies of tall red. Here, we again selected two plants, one is male plant and one is female plant, and carry out the cross between those two plant. For the phenotypic character, the dominant character is tall and red, that means tall height and red color flower, or which we called purple color flower, and dwarf and white, which is the recessive characters. When we see the genotype of this is capital T, capital T, capital R, capital R, and in case of dwarf plant, small t, small t, small r, small r. The gametes of parent is capital T, capital R, and the second parent, that is recessive parent, is small t, small r. When we fuse that gamete together and after fertilization, the resultant F1 generation is with capital T, small t, capital R, small r type of genotype, and all the progenies which shows such a type of genotypes are with tall height and red color flower. When we carry out selfing of F1 generation, like capital T small t capital R small r is considered as a female plant, and same genotypic plant when we cross with male plant, the gametes of F1 we will get capital T capital R, capital T small t capital R, and small t small r. Then the F2 generation is with, when we show the Punnett checkerboard, when we cross those gamete, then we will get 9 tall red plant, 3 tall white plant, 3 dwarf red plant, 1 dwarf white plant, and the phenotypic ratio is with 9:2, 3:2, 3:1. Here the law that shows the expression of tall character with red character does not influence the, that means the color of that means the character which dominant for color never mask the expression of height character, that is the capital T and capital R, they does not influence their phenotypic expression. Thank you.